Hello, students. Module 7, Lesson 5. This is our last lesson in Module 7. We will be adjusting for multiple hypothesis testing. Now, as with a few other lessons in this module, I'm going to do an overview and short lecture all in one video. So, what we are discussing is when you have many hypothesis tests on the same data set, and we need to correct for that with an adjustment. The objectives of this lesson are to correct for multiple hypotheses. We will discuss two methods, a Bonferroni correction as well as a Benjamini-Hochberg correction. The second objective is really to be able to conduct hypothesis testing with a controlled false discovery rate. Okay, well, let's get into my short lecture. Often, I will highlight this word, often experiments in bioinformatics involve a huge number of statistical hypotheses tested on the same data set. Could be hundreds of thousands. And even if you have a small significance level, you will have many false positives because of the sheer number of hypotheses tested. Just as an example, suppose we have 1,000 true null hypotheses, and we have an alpha equals 0.05 significance level. Well, on average, you see we can multiply. We would have 1,000 times 0.05, which is 50 false positives. Or we know these are type 1 errors. And this is a lot. To control the rate of these false discoveries, we need to adjust for our multiple hypotheses being tested. And I mentioned when I was talking about objectives, there's really two methods we will discuss. The first is a Bonferroni correction, and the second is a Benjamini-Hochberg correction. As an example from my own experience in correcting for multiple hypothesis tests, ChIP-seq is an assay to measure protein binding sites on the genome. It is one of the many sequence synthesis methods that involve sequencing short fragments of DNA, mapping them to the genome, and then using that to make some sort of biological conclusion. In ChIP-seq, or the data analysis involved in ChIP-seq, we must decide what are so-called peaks in a coverage curve. Now, when I was a postdoc at Berkeley, we developed an algorithm called ChIP-seq, and we used a Benjamini-Hochberg correction, which is one of them that we will discuss. Let me just show you our paper. Here is our paper published in BMC Bioinformatics. You can see you can access it for free. It's called Shape-Based Peak Identification for ChIP-seq. Now, I'm not going to get into the algorithm or really even any more details about the ChIP-seq assay, but what I will click to is here, this paragraph where we talk about correcting for multiple hypotheses. You see, this is relevant to this lesson. In fact, this is what we're learning about in this lesson. You see, we use a Benjamini-Hochberg correction, and then we just explain how this correction goes, which is how Benjamini-Hochberg goes in general. We order the p-values, and then there's a way to make a cutoff where you reject some of the null hypotheses and not others. We will use R for this Benjamini-Hochberg correction. So I just wanted to show this so that you understand its fundamental importance in biological and bioinformatics research. Okay, back to my iPad. As a remark, when we ran ChIP-seq on some publicly available data, this is fruit fly data, ChIP-seq averaged over 8,000 statistically significant peaks, meaning protein binding sites. So Imagine that is the number that's statistically significant. There are so many more hypothesis tests being run here. I think also when we ran this on human data, we had about 50,000 statistically significant. So correcting for multiple hypotheses is quite important. Okay, now let's get into a little bit of these two types of corrections. Suppose we are testing M, I'll write this, we have M null hypotheses. M0 is the number of true null hypotheses. R is the number of rejected null hypotheses. So what do we know here? We know this number and we know this number. We don't know M0. And we can make this table, which is kind of like one we discussed when we were first discussing hypothesis tests, where maybe the null is true, maybe the alternative is true, and then maybe we reject and maybe we don't. So altogether, we have M hypothesis tests. We reject R of them. So these two numbers we know, but pretty much everything else we do not. And V is the number of type 1 errors. That's the thing we're worried about having too many of because we have so many hypothesis tests. So, as I was mentioning, U, T, V, S, well, we cannot observe any of these. In the Bonferroni adjustment, we just reject all null hypotheses with p-value less than alpha over m. So if you have m null hypotheses, significant level alpha, we just divide alpha over m, and this is kind of a new cutoff. Now this controls what's called family-wise error rate. It's the probability of at least one false positive, or the probability of at least one type 1 error. This family-wise error rate is the probability that this V is greater than or equal to 1. Now, Benjamini-Hochberg controls for what's called a false discovery rate. This is the expected number of type 1 errors among the rejected hypotheses. The expected number of type 1 errors among the rejected hypotheses, with the convention that if you have 0 rejected hypotheses, the FDR is just 0. Now, Bonferroni is quite strict, and because of this, we have very low power. Family-wise error rate controls the false discovery proportion in all hypotheses tested, whereas false discovery rate controls only the proportion in the rejected hypotheses. And so this false discovery rate, it is less strict, and generally there's an inequality that the false discovery rate is less than or equal to the family-wise error rate. So if you are putting a bound on this, it's not going to be as strict as putting a bound on family-wise error rate. Controlling this false discovery rate, which is what Benjamini-Hochberg method does, leads to more powerful tests. That's why we used it in our paper. And you're able to make more discoveries. So for us, as I mentioned, we will use R. We can use this function p-adjust. Now, one thing that I want to mention here is we have what's called p-values and we have what's called q-values. So the outputs of this p-adjust are called adjusted p-values. If you are doing a Bonferroni correction, the output will just be m times this p-value that you originated with. This leads to a bound on the family-wise type 1 error rate. And so this is truly an adjusted p-value. You are just adjusting the original p-value. However, the Benjamini-Hochberg correction does not control the type 1 error rate. It's not the goal. Instead, it controls the false discovery rate. And so the q-value is not exactly a p-value. So this, the name, when you are doing this one, the name adjusted p-value. If you are working with Benjamini-Hochberg, it's a bit of a misnomer. Misnomer. My final comment here in this short lecture is the following. If you use this method, FDR, or another way to say that is the Benjamini-Hochberg correction, it changes the p-value of an individual test to q-value. This q-value, we don't necessarily need to stick to the 0.05 level, which is conventional when you are discussing p-values. And then I make a remark, if you have sparse discoveries, sometimes bigger q-values make practical sense, whereas a p-value of 0.5 would not make practical sense. This is all I will say about the Bonferroni and Benjamini-Hochberg corrections at the moment. At this point, you should work through our Canvas pages where we have some R commands to practice this p-adjust. Moreover, one of the things you'll see is a beautiful Monte Carlo study where we look at different methods for correcting multiple hypotheses. It's quite fun. Thank you so much, class.